Hello and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Turi King, your host, and today I'm talking to Robert Kelsh, who is Professor of Stem Cell and Developmental Genetics. His research focuses around fundamental questions in developmental biology. So, Robert, I love to hear how people came to be interested in the field that they're in. So, first of all, for people who don't know, what is developmental biology and how did you come to be interested in it?
Developmental biology is the process of understanding how a fertilized egg becomes a functional organism. So I study basically stages of development in fish that are the process from very early fertilization of the egg all the way through to a functioning larva. I don't usually look much beyond about 5 days of development, but by that point a zebrafish is essentially ready to be free living and fully functional. I'm a very broad biologist at heart, and developmental biology brought together genetics, anatomy, biochemistry, all sorts of things come together. So it's a very sort of integrative discipline. And by the end of my third year of study, I realised that that was where I wanted to go. Up until then, I had been quite polarised. I'm actually, as you well know, a very keen natural historian. And so for a long time, I was trying to decide between some sort of bird ecology type thing and developmental biology. But developmental biology won out because I thought the questions were more exciting, more open, it was less known.
And they're some of the most fundamental questions we have kind of about life on Earth. It's like, how does something develop, going from like a single cell to, right, well, the head's up here and the legs are down there and the arms are in the middle type thing.
Yep.
And that's basically what you're trying to understand, isn't it?
Yes. I mean, my own focus has been on two questions. So the two questions are, how do you make pigment cell types? There are 3 different colours of pigment cells in zebrafish. And I was interested in how you make those 3 cell types. So that's a classic problem in developmental biology. How do you make different cells to make different structures of the body? And then the second classic problem is the patterning problem. How do you put them in the right place? Because if you don't put them in the right place, you end up with a horrible mess. So we've worked mostly on how you make pigment cells, and then we've worked on how you pattern those cells. And the beauty of the patterning thing is that in zebrafish, the adults have this beautiful longitudinal stripe pattern, right? Very bold, very colourful, very pretty. And it's a lovely situation where you can ask, how do you disrupt that? And there are many groups that have done a lot of work looking at identifying genes that change that pattern when mutated. And we have got involved in looking at that from a more hypothetical perspective. So those are the two questions. How do you make different cell types, and how do you pattern them in the correct way? And actually the pigments, you may think, well, again, why pigment cells? We don't care how you make different types of pigment cells, but actually you should, because the pigment cells come from a set of cells that are essentially a set of stem cells. And those stem cells are capable of forming an incredible diversity of cell types. And the pigment cells are a subset of those cell types and have been considered to be a coherent group, a related group of cell types. I consider them to be a model within a model for how this stem cell population, which are called neural crest cells, how they make different types of cells. And that full repertoire includes many, many types of neurons, neuronal support cells called glia, all sorts of other things as well. The neural crest cells are really important. The pigment cells are an important derivative of those crest cells. But also they're a very visible derivative. And therefore for these screens, which were done entirely visually, they're a fantastically accessible model system. So I consider them a model within a model for neural crest stem cell development. And that's why we study them.
So this is coming back to model organisms. So for people who don't know what a model organism is, what's a model organism? What makes good genetics? What makes bad genetics?
Yes. So a model organism is an organism that scientists have, for whatever reason, chosen to bring usually into the lab and study. And for different disciplines, you would use organisms with different characteristics. But if you're a geneticist, you want an organism that's easy to work with from a genetic point of view. So the key characteristics would be that ideally you'd have a short generation time. They'd breed rapidly, they'd grow rapidly, and then they'd become fertile adults quickly. You would need an organism where you can encourage the formation of mutations, so that's the ability to damage the DNA in a way that's easy to apply.
And why do you want the mutations?
Because by default, really, the way you study things is by asking what happens when you mess it up. So you want to know what a gene does. Well, the best way of doing that is to say, okay, here's the organism. This one's a wild type. This is developing with that gene functioning. And here's exactly the same organism, but where we've mutated that gene. So a very obvious example is tyrosinase, right? So tyrosinase is an enzyme, a protein that helps you make black pigment. And so if you have a mutation in tyrosinase, you fail to produce black pigment, or you don't make as much. So you get an albino phenotype. And many of you will be familiar with that in a human context. But it's true in all sorts of animals as well. I mean, zebrafish have albinos, fruit flies have albinos, in fact, for that matter. And that's because they all have the tyrosinase gene, and it has the same function in all of these organisms. It's used to make melanin biochemically. And if you mutate it, you get the same effect. You don't have melanin, right? So the best way to study what a gene does is to ask what happens when you mess it up. So you need to be able to make things go wrong easily in order that you can generate the mutants that you want in order to study them.
The other thing about model organisms as well, isn't it, is that if they have a short generation time and they have lots of offspring, you can see stuff happening much more quickly than if you did it with us humans who have very long generation times. Yes. And also, you need the numbers as well, don't you?
You do. So when you mate a pair of zebrafish, you will get hundreds, literally hundreds of fertilized eggs. And genetics is all about the numbers. So there is a lot of classical theory around the patterns of inheritance, which means that you want to count the numbers of offspring that have an aberration. There's some classic ratios that you expect. And so if you've got 5 embryos, or 6 or 7 or 8, which is what you would get even with something like a mouse, which is a very good model mammal for doing genetics, but compared with a zebrafish, in terms of numbers, is rather awkward. With zebrafish, you'd have hundreds of offspring. So if you're looking for a special ratio, 25% is one of the numbers you're often looking for. If you're looking for 25%, 25% of 100 is, of course, 25, and there's a bit of variation, so you might expect 23, 25, 27, whatever, but you know whether it's a quarter. If you've got 5 embryos and you're looking for 25%, you're hoping to see 1. And so that just makes the statistics much better if you've got these large families. So the zebrafish from that point of view is really very good indeed.
And you're doing something where, I mean, this is one of the really big questions, isn't it? How on earth do our cells know you're going to be a skin cell and you're going to be a brain cell and you're going to be a kidney cell? And the way that you are looking at that is by looking at a subset of cells, the pigmentation cells, to try and go, well, if these ones know which color they're supposed to be and also where they're supposed to be, because that's another big question, you're using that to answer kind of some of the bigger questions about how cells know what they are and where they're supposed to be.
I told you that neural crest cells can form all these different cell types. Neural crest cells are actually fascinating in a number of ways, but one of the things that is important, in order that they form all these different cell types that do different things, it's almost inevitable that they have to go to different places. So the neural crest cells form actually from a very early stage of the developing spinal cord. Again, at this very early stage, they emerge from the developing spinal cord and they will migrate through the body. So the pigment cells are in the skin. Most of the neurons are rather deep inside the body in clusters called ganglia. There are skeletal cells, cartilage cells that form and contribute to the face that are derived from the neural crest. So, there's all sorts of different types of cells doing different things and therefore having to be in different places to work properly. So they're highly migratory. Neural crest cells as a population are highly migratory. So you have this situation where these cells are formed from a relatively well-defined position close to the spinal cord, but then disperse through the body. And as they disperse, they navigate to different locations. And there's this big question of when and where do they learn what they are? Do they know what they are and therefore migrate to the right places because they know where they want to be going? Or do they migrate to the right places and then locally get told what they should be according to where they are? That actually was one of the big questions underlying one of the main topics that we have been trying to address. at the core of making a cell different to another cell is the expression of a special set of genes that encode proteins called transcription factors. And the reason that that's the way it works is because transcription factors are proteins that activate or shut down genes. So if you want to make a black pigment cell, a melanocyte, you need to turn on all the genes that make a melanocyte. And that includes tyrosinase, but it includes a whole load of other genes that do the biochemistry of melanocytes, but also genes that do the cell migration of melanocytes, and genes that do all the other things that melanocytes need in order to make a black pigmented cell that is in the right place, in the skin or wherever else. So fundamentally, how you make different cell types can almost be reduced to how do you turn on the right transcription factors in the right places. So the set of transcription factors that a cell expresses, the ones that are active in that cell, they determine what that cell becomes. And so for the pigment cells, for example, we know of key transcription factors, and actually there are surprisingly few perhaps. There are key transcription factors where if you mutate one of those genes and you lose the function of one of those proteins, you now essentially shut down completely the possibility of becoming a melanocyte or of becoming one of the other cell types. There's a lovely shiny silver cell called an iridophore, and there's a yellow cell called a xanthophore. And if you mutate individual transcription factor genes, you can't make a melanocyte and you can't make an iridophore. And what we've recently discovered is that if you mutate both of them, so you have both of them inactive in crest cells, then you can't make the xanthophores either. And that was something that was, when I started looking at this, that wasn't expected. They seem to work together somehow to make the yellow cell, the xanthophore. And that's slightly interesting, really, because here's a protein that controls the activity of genes to make a melanocyte, turn on tyrosinase and so on. And then you've got another transcription factor, which turns out, in fact, to be a very closely related type of protein. And that is required to make a shiny silver cell. And a shiny silver cell really does look very different to a black melanocyte. You wouldn't have any trouble seeing the difference if I showed you them down the microscope. But if you mutate one, you lose melanocytes. Mutate the other, you lose iridophores. But if you mutate both together, you lose melanocytes, as you expect. You lose the iridophores, as you expect. But interestingly, you now suddenly lose the xanthophores as well. So you don't have yellow cells. And I guess what, in my mind at least, the protein that does this for melanocytes was one of the classic examples of a master regulator. It was thought that its role was always to make a melanocyte. And I suppose what I think we are learning from this recent discovery is that, yes, it's crucial for making melanocytes, but actually it also, in other contexts, can be crucial for doing something else as well. It's just that it works there alongside another protein that is, if you like, equally a master regulator. And so it's only when both of those are now gone that you see the effect.
So one of the interesting things that's coming across from your work is not only is it kind of looking at really sort of fundamental how things develop, but I'm guessing it's the sort of thing that the reason why you're looking at it in a model organism is because that information can be used for other things. So what is it that it could be used for in future? Is it something that could be used in medicine?
Yes, so I'm always slightly hesitant about the application question. You know, it's appropriate that we do justify what we're doing, but I have to confess that I have always had the view that looking for curiosity's sake is a legitimate reason. And so my own work, my lab's work, has been very much motivated by wanting to understand the process. How do you make cells different? How do you pattern the cells in the right way? I just want to understand the biology of it. These are, I think, very beautiful questions. There is application, of course. So in human context, understanding how melanocytes are made and how melanocyte development is controlled is very important. There are, of course, many human conditions where melanocyte development goes wrong. Albinism we've mentioned, vitiligo is another. There are lots of different conditions that are affected this way. And of course, there are melanomas associated with these pigment cells. So in all of those ways, melanocytes are important, and understanding melanocyte genetics is important. And the only thing that makes me slightly hesitant about that argument is that in the context of development, when these things go wrong, they go wrong very early in the embryo. That's an embryo that in a human context is in the mother. Some of this is happening at very early stages, probably before you even know you're pregnant. And many of the mutations that cause these defects in humans are spontaneous. So I told you in the fish, when we're studying these things, we actively have to treat the embryos, or the parents actually, with a mutagen, a compound that will damage DNA, in order to create the mutations so that we can find them and study them. Obviously, that's not something that you do in humans, but in most cases, these conditions come about through spontaneous mutation. And so it's not like you can say, oh, well, the mother was affected or the father was affected or both were affected, and therefore you know that the offspring are likely to be affected. That's usually not the situation. And so if you don't know which individuals are at risk, doing something about it is very difficult financially. Do you screen all fertilized embryos? And ethically as well, would you even want to do that? So there are some conditions where the onset is late enough that, of course, you could intervene in a focused way. And there are even some pigment cell conditions where that's the case. Vitiligo, for example, is a late onset thing. But I'm hesitant to push too hard the idea of treatment. However, what I would say is that one of the genes that we have studied most is a gene called SOX10, and SOX10 is associated with human congenital defects of pigmentation and of nervous system development. And some years ago, I had the privilege of talking to parents of patients, actually. And I realized that for them, actually understanding the child's condition has value. So even if I can't offer a treatment for the SOX10 associated conditions, what I can offer is an understanding of why their child has the problems that that child has. And actually, I think that is very important. And if you want to apply an application, that's the justification for why we ask the questions we ask. The fundamental understanding that we're aiming for gives understanding of the situation when it doesn't work correctly, and that affects people. And it's helpful for those individuals to have some understanding of what has gone wrong.
So what's the end goal of your research?
So the end goal is to understand how these processes work. So these two transcription factors that work together to make the yellow cell, right? A black cell transcription factor, a shiny cell transcription factor work together to make a yellow cell. So I would like to know, how does that work? Do those two transcription factors work side by side, binding to the same genes and turning them on? I want to know whether it is those two key target transcription factors downstream of them that I think is what is important. I want to know whether that's really all it is. I want to know whether those two transcription factors, the black cell one and the shiny cell one, whether they continue to be needed. It could be that they turn on these other key transcription factors for xanthophores, but then they're also required as an ongoing process to contribute, working probably alongside those target transcription factors. I'd like to understand it at that level. I don't think, it certainly won't happen in my career, that I will understand every single gene interaction process for those 4 transcription factors in those different cells. And I don't think I need to understand that, actually, because I'm more interested in the principle of how it works than every single nitty-gritty detail.
So why do you think understanding evolutionary processes is so important? We are in the Milner Centre for Evolution. Why do you think it's so important to understand evolution?
Well, okay, that's very straightforward. So I love the diversity of nature. I love the details of what makes things look different. And that's come out of evolution. In a research context, actually, I have a very specific interest, and that's about how you evolve different types of cells. So mammals, including humans, only have one type of pigment cell. They have the melanocyte. And Well, zebrafish has 3. Actually, probably they have 4, but one of them is a small component in adults only. But there are other fish that have more. So there's another fish species that we've worked on. It's a Japanese fish called Medaka, Japanese rice fish. And we have worked with Japanese Medaka scientists to try and understand the same question, how you make pigment cells in Medaka. And part of the motivation there was the fact that there are 4 types of pigment cells in Medaka, not just 3. So how do you make that extra cell? But what I've become even more interested in recently is the evolution of novelty, or rather how that might come about. So in terms of melanocytes, how do you evolve a melanocyte, a cell that makes melanin? I've already told you that actually many organisms have tyrosinase. Actually, that even goes to things like fungi and stuff. So making melanin as a compound, a black pigment, is an ancient thing. But a melanocyte is a bit more than that. A melanocyte in mammals is a cell capable of making melanin, but it makes melanin in little packets. They're called organelles, little subcellular structures that are basically like little balls with a wrapping membrane. Think of it as a sort of rugby ball that you've injected black stuff inside, but very tiny. So there's all the process of making that structure and putting melanin in it and not allowing melanin to be elsewhere. So that's, in a vertebrate context, in a fish or in a mammal, that's what a melanocyte is, roughly. Now, there are other places where you can make melanin, but usually in a more loose way. And so you can kind of imagine that to evolve a melanocyte, you had the biochemistry, but now you had to evolve the way to package it up within a bit of the cell and make that work, right? But there are other pigment cell types in other fish, not ones we've studied. If you think about tropical reef fish, it doesn't take much to remind yourself of just how colourful they are. And actually a lot of that colour can probably be explained by the 4 pigment cells that Medaka have. But there are definitely situations where there are other pigment cells. There's a lovely thing called a cyanophore. It's a very blue cell, intensely blue. It's found in a rather small subset of fish that have intense blue coloration. And last time I looked, we had no real idea what this pigment was, except that it was definitely blue and it was different. So this is a novel pigment cell type that we know nothing really about. We don't know anything about the genetics, we don't really know anything about the biochemistry, and it seems to be found only in a few fish. So how on earth did those or their ancestors invent a new pigment cell? And it's probably not the only example. So the other 4 types, they are black, shiny silver, yellow, and a sort of whitish cell. I wouldn't be surprised if amongst the diversity of fish, if there are multiple ways that fish have evolved to make a shiny silver cell, for example. Maybe what we think of as one cell type, an iridophore, is actually 2 or 3 different cell types with different underlying biochemistry that we just don't realise because we haven't looked closely enough. So I would love to spend some time looking at that diversity, trying to identify new cell types, and then trying to understand something of the genetics that underlies that. And then behind that, of course, is the evolutionary question of how did that new cell type evolve?
Robert, thank you so much for talking with me. This was a podcast by the Milner Centre for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us via our social media. For more information about the Milner Centre for Evolution, you can visit our website.